Hello and welcome. This video is for AQA A-Level Biology Unit 4. It's worth watching the Unit 1 video for DNA structure before you watch this, or at least doing a recap about DNA structure. Other than that, there's timestamps below in the description for easy navigation. We start off with DNA, genes, and chromosomes. DNA, genes, and chromosomes. We'll start by taking a look at eukaryote and prokaryote DNA. Eukaryotes first. These are plants and animals. DNA in eukaryotes is in a nucleus. It's very long, linear, and associated with proteins called histones. Here is a molecule of DNA. Here are our histones, which are spherical proteins, and the DNA is associated with those histones. It can then be further packaged and arranged to make chromosomes. A DNA molecule and proteins, the histones, form a chromosome. Prokaryotes. DNA in prokaryotes is short, circular, and not associated with histones. Here's a prokaryote, a bacterium. You can see the main DNA in the center of the cell there. Prokaryotes may also have plasmids, which are small rings of DNA separate from the circular DNA. Here's a plasmid. Next, mitochondria and chloroplast. Here is a chloroplast, here is a mitochondrion. Mitochondria and chloroplasts have their own DNA and their own ribosomes. Their DNA codes for proteins, for example enzymes, and in mitochondria the enzymes are for respiration, and in chloroplasts the enzymes are for photosynthesis. We'll take a look at that in more detail in Unit 5. Like prokaryotes, the DNA is short, circular, and not associated with histones. You should be able to make a comparison of the arrangement of DNA in eukaryotes, prokaryotes, and compare that to that in mitochondria and chloroplasts. Genes and DNA. Here is a molecule of DNA again. We can magnify a region of it and you can see it's in its double helix structure. And you can imagine that along that DNA we have a gene. A gene is a base sequence of DNA that codes for the amino acid sequence of a polypeptide. In other words, the correct order of amino acids in a polypeptide, the primary structure, or a functional RNA. For example, ribosomal RNA, which makes ribosomes, and tRNA, which helps with protein synthesis. Students often forget the second function of a gene there, so it's worth making a note and making an effort to remember that. Genes and DNA, let's have a look at that in a bit more detail. A gene occupies a fixed position called a locus on a particular DNA molecule. The same gene is always found at the same locus. A locus is a bit like an address for the gene. Triplets code for specific amino acids. A triplet is a sequence of 3 bases. Here's a DNA molecule. Let's separate out the strands so we can see a base sequence, and here we have some bases in the DNA, and we can see that there are 1, 2, 3 triplets. Many triplets together, the genetic code for an amino acid sequence. For example, here's a diagram to show several amino acids in a sequence. The genetic code is universal. This means the same triplets always code for the same amino acids in all living things. For example, the DNA triplet TAC always codes for the amino acid methionine. We don't need to memorize specific triplets or specific amino acids. This is just to illustrate the idea that DNA is universal. DNA is also non-overlapping. Each base is only part of one triplet. Adjacent triplets do not overlap. So in our diagram there, we don't get triplets like this. That's not how they are arranged. DNA is also degenerate. More than one triplet can code for the same amino acid. For example, the DNA triplet GTT and GTC both code for glutamine. Again, we don't need to memorize triplets and amino acids. This is again just to illustrate the idea that DNA is degenerate. Next, we take a look at protein synthesis. Before we do that, we need to understand the structure of DNA a little bit more. DNA has coding and non-coding regions. Coding DNA codes for polypeptides. But most of eukaryotic DNA does not code for polypeptides. Most is non-coding. There are many non-coding regions within and between genes. So here is a molecule of DNA, or part of a molecule of DNA. Let's imagine we've got two genes. We'll call them gene 1 and gene 2. And in gene 1 we have coding regions, and coding regions are called exons. So there's two coding regions there, two exons. And we also have non-coding DNA within the gene, and that is called an intron. Same again for the one below. We have exons and we have introns. Worth noting that there are usually several introns between exons, not just one as shown in my diagrams. There is non-coding DNA between genes. This often contains sequences, repeat sequences of bases. For example, that repeat sequence there. Also worth noting that non-coding DNA also has regions that regulate genes. We'll look at that in more detail in Year 2 A-Level Biology. Genomes and proteomes. A cell contains genes in its DNA. The complete set of genes a cell has is called its genome. DNA in a cell codes for proteins. The genes code for proteins. The full range of proteins that a cell can produce is called its proteome. Two important definitions there for genome and proteome. Protein synthesis. Let's have a look at an overview of protein synthesis. First, we have transcription. This happens in the nucleus of a cell. And that leads on to translation. This happens in the cytoplasm on ribosomes. This produces polypeptides. So this is protein synthesis, transcription and translation. We do also have folding and modification of those polypeptides, and that's done in rough ER and Golgi. So this is processing. Proteins are then secreted or used within the cell. Two important molecules that we need to know in protein synthesis before we get into the details of it, they are messenger RNA, otherwise known as mRNA, and transfer RNA, otherwise known as tRNA. Messenger RNA is linear. In transfer RNA, it's clover-shaped. There are no hydrogen bonds in the linear structure of mRNA, and transfer RNA actually has hydrogen bonds between mRNA strands. You can see the hydrogen bonds there in red in the diagram. Messenger RNA has codons. Here's a codon in the messenger RNA. And transfer RNA has anticodons. The anticodon is shown there. There are no amino acid binding sites in messenger RNA, whereas transfer RNA has an amino acid binding site on each tRNA molecule, as shown there at the back end of the tRNA molecule. So that's an overview of the process and some key definitions and some key molecules. Next, we're going to take a look at an animated version of protein synthesis, followed by a point-by-point list description of how protein synthesis happens. So let's zoom into the nucleus there and look at a molecule of DNA, or part of a molecule of DNA. We can separate the strands in DNA, which is the first step. We have our base sequences there that's shown on what we call the template strand, and we have free nucleotides in the nucleus. We will have base pairing, T with A, but remember, uracil replaces thymine in mRNA, so adenine base pairs with uracil instead of thymine. A will base pair with U. So next we have U, and then we have the involvement of a molecule, an enzyme called RNA polymerase. This catalyzes the formation of a phosphodiester bond between adjacent nucleotides. We have a continuation of base pairing and the formation of phosphodiester bonds, and in this way we form or produce a copy of the DNA strand in the form of a messenger RNA molecule. However, this molecule is actually referred to as pre-mRNA. It still contains introns. We then have the process of splicing. Splicing removes the introns, and then we are left with mRNA. The mRNA leaves the nucleus via a nuclear pore and goes into the cytoplasm. Here, it will join with the ribosome. So there's our ribosome, there's our mRNA. The mRNA will join the ribosome. We then have the involvement of our tRNA molecules, each with an amino acid bound to the amino acid binding site. So there's our tRNA with the amino acids attached. The codon on the mRNA will base pair with the anticodon on the tRNA. This brings our first amino acid to the mRNA. We then have another codon, complementary base pairing with an anticodon on tRNA, and that brings two amino acids together, and we have the formation of a peptide bond formed via a condensation reaction. Very important to remember, we have the use of an ATP molecule to release energy for the formation of that peptide bond. This process then continues. Codons join with anticodons and new amino acids are brought along. Peptide bonds are formed. Each time a tRNA molecule is released after it has delivered the amino acid, and we go on to form a polypeptide. So there is our polypeptide. That was the process of translation. So let's go through the steps outlining the key detail that you need for protein synthesis. Protein synthesis, step 1, we have transcription. This happens in the nucleus of a cell. Hydrogen bonds between DNA bases break to expose the template strand. One DNA strand acts as a template for the formation of mRNA. You can see the template strand there. Free nucleotides align by complementary base pairing. Remember, to make mRNA, uracil is used in place of thymine. RNA polymerase joins nucleotides with phosphodiester bonds between adjacent nucleotides. Pre-mRNA is made. Introns are removed. Pre-mRNA is spliced to form mRNA. So there's our pre-mRNA, we have splicing, and then we have our mRNA. mRNA exits the nucleus via a nuclear pore. Then we have translation. This happens in the cytoplasm. mRNA attaches to a free ribosome or a ribosome on rough endoplasmic reticulum. Then we have tRNA, which brings a specific amino acid, and the tRNA anticodons bind to the complementary mRNA codons. Amino acids join by condensation reactions to form peptide bonds, and remember, this is with the use of ATP for energy. tRNA is released after the amino acid joins the polypeptide, and the ribosome moves along the mRNA to form the polypeptide. So these are the key details of transcription and translation in protein synthesis. Next, we take a look at mutations. Mutations. Gene mutations involve a change in the base sequence of DNA. These mutations occur during DNA replication. They happen spontaneously at any time, and they happen randomly at any place in the DNA. But the rate of mutation, or rate of gene mutation, how often they happen, can be increased by mutagenic agents, for example, radiation. Important to note that mutagenic agents, for example, radiation, don't cause mutations, they increase the rate of mutations. So here is a base sequence with no mutation. We can see that there are 4 triplets, so that's going to code for 4 amino acids. And here are the 4 amino acids shown by different shapes. A short part of a polypeptide. This will form a polypeptide, so there's our amino acid sequence. This leads to the production of a polypeptide and a functional protein. For example, an enzyme. And here is a simple diagram of a functional protein, for example, an enzyme. The first mutation is a base substitution. This is where one base is replaced by another. Here's our original base sequence in DNA. We can see that a C, a base sequence of C, cytosine, is replaced by G, guanine. The triplet base CGA is now GGA. This could code for a different amino acid. So here's our original base sequence, and here's our new base sequence. One amino acid has been changed. Remember though, the new triplet could code for the same amino acid because the code is degenerate. This means more than one triplet can code for the same amino acid. However, there are various things that can happen as a result of a base substitution mutation. So here's our structure of our original protein. If a mutation codes for the same amino acid, the result is we're going to have a new protein that is exactly the same as the original protein. There's no change. If it codes for a different amino acid in a non-crucial position, we might have a slight change in the structure of the protein, for example, in that little area there, but overall it doesn't change the performance or the action of that protein. We could have a different amino acid in a crucial position, for example, affecting the type of bonding that goes on, and that could have a significant change on the protein. Base deletion. A base is deleted. So here's our original base sequence again. The C has been deleted. Deleting a base changes the entire base sequence after the deletion point, everywhere from the deletion point onwards. This is a frame shift mutation. So this is the original sequence of amino acids, and this is the new sequence. We can see that every amino acid after the mutation point has changed. All amino acids could be altered after the mutation point, therefore changing the primary structure of the protein. This makes a significant change to the amino acid sequence and therefore the tertiary structure of the protein. Here we have our original protein and the new protein. As you can see, there's a significant change in the final shape due to a significant change in the amino acid sequence. We also have chromosome mutations. Mutations in chromosome number can happen by chromosome non-disjunction. This is when chromosomes fail to separate properly during meiosis, leading to gametes with too many or too few chromosomes. An example is Down syndrome, where one gamete has an extra chromosome. So that's an overview of mutations and their effects. Next, we take a look at meiosis. Meiosis. This is a type of cell division that produces gametes, in other words, sex cells. This occurs in reproductive organs, for example, testes and ovaries. Here's an overview. We have a cell with 46 chromosomes. We go through meiosis and we end up with gametes, sperm cells and egg cells, each with 23 chromosomes. This example is for humans. Meiosis halves the chromosome number in cells. The diploid number is the full set of chromosomes, so we refer to that often as 2n. And the haploid number is a half set of chromosomes. We refer to that as n. In humans, the haploid number is 23, therefore the diploid number of 2n is 46. An example of a housefly has a diploid number of 12 and a haploid number, therefore, of 6. For rice plants, we have a haploid number of 12 and therefore a diploid number of 24. Fertilization combines two haploid gametes, restoring the diploid chromosome number. Here are two gametes joining, each with a haploid number of chromosomes. The gametes join and form a fertilized egg with 2n, in other words, a diploid number of chromosomes. Steps in meiosis. Before meiosis begins, we have the replication of DNA. And as a first step in meiosis, we often talk about the formation of chromosomes. This is 2n, this is diploid. Chromosomes are made of two sister chromatids, and there are 23 pairs of chromosomes. The pairs are matched in the genes that they contain. However, they may have different versions of the genes called alleles. The matching pairs are called homologous pairs. The chromosomes line up as homologous pairs. We still have a diploid number or 2n number of chromosomes. We then have the first meiotic division. Homologous chromosomes are separated and the chromosome number is halved. So now each cell has an n number of chromosomes. We then have the second meiotic division. Each cell has a haploid number of chromosomes. 4 haploid gametes are formed. each is genetically different due to the variation in alleles. For example, if we're talking about the gene for blood group, we may have an allele for blood group A or for blood group B. Just worth mentioning here, going back to our idea of non-disjunction, here is an example where we have non-disjunction. Chromatids do not separate, so we have one cell that has an extra chromosome. Normally though, this is what happens. So this is an overview of meiosis and the steps in meiosis. Meiosis and variation. Meiosis can contribute to variations in individuals. First thing is independent segregation. Here we have two pairs of chromosomes aligned in their homologous pairs. On one side we have the paternal chromosomes that are given by sperm cells from the father, and here we have maternal chromosomes given by chromosomes from the egg cell from the mother. When homologous chromosomes line up, they don't always align in the same way. So here's an enlarged version of the diagram above. Here's one arrangement of the homologous chromosomes. Another arrangement would be like this. We can also have an arrangement like this. So if we take a look, there's actually 4 ways in which these homologous chromosomes can align themselves. This process is called independent segregation. 2 pairs of chromosomes have 4 ways to line up. We can work this out by using 2 to the power n, where n is the number of pairs of chromosomes. So we have 2 pairs of chromosomes. We would work this out by 2 to the power of 2, and that gives us 4. In humans, we have 23 pairs of chromosomes, so we would work that out by saying, or calculating, 2 to the power of 23. That gives us over 8 million ways of the chromosomes lining up. That's a lot of possible variation. One more example, if we had 20 chromosomes, that's 10 pairs, we would use 2 to the power of 10. That gives us 1024 ways of those chromosomes lining up. This is independent segregation. We can also have crossing over. Here are 2 homologous chromosomes. One is paternal, one is maternal. They are homologous. And here, in the region where we have the letters A and B, that's the locus or the position for the gene for blood group. It has 2 alleles. The alleles are capital A and capital B. When homologous chromosomes line up, parts of the chromosomes can switch places. The points where they switch are called chiasmata. If you have one of those, it's called a chiasma. So here's our chiasma on our diagram. This is called crossing over. The alleles can change chromosome. So we can see what happened there. Parts of the chromosome have switched, the alleles have switched places. Crossing over can happen at 1 to 3 points per homologous pair. This results in 23 to 69 crossing over points per division. This increases variation. So that's crossing over. We also have the idea of random fertilization. This is after the gametes have been formed. Any sperm cell can fertilize any egg cell. Here are just 4 sperm cells and 4 potential egg cells, but remember, there's potentially millions of sperm cells and potentially thousands of egg cells. This sperm cell could join any one of those egg cells, and this sperm cell could also join any one of those egg cells. Only one sperm cell will join one egg cell. Any one egg cell out of thousands could meet any one sperm cell out of millions. This process is known as random fertilization. Random fertilization introduces another layer of variation. If you were asked how meiosis contributes to variation, we can simply summarize it like this. Meiosis leads to variation in offspring by 1. Independent segregation of homologous chromosomes. This means maternal and paternal chromosomes are reshuffled in any combination. Crossing over. This is the exchange of segments of non-sister chromatids between homologous chromosomes. Additional variation is introduced by random fertilization, as any sperm cell from the father can fertilize any egg cell from the mother. So this is meiosis and how it contributes to variation. Next, we're going to take a look at genetic diversity and adaptation. Genetic diversity and adaptation. Genes code for specific characteristics. Genes can exist as different versions called alleles. Let's take a look at an example of how this works. Here we have a population of mice. And let's take a look at an example of the gene for fur color, which they all have. There are different versions of the gene for fur color called alleles, and these alleles could be brown or grey or white. One more example, the gene for tail length, and the alleles for that could be long or short. So that's two examples of genes and their alleles. Genetic diversity, this is a definition that we need to know and remember. Genetic diversity is the number of different alleles of genes in a population. Let's explain genetic diversity a bit more. For example, there are two populations of mice of the same species but living in two different areas. A mouse has about 20,000 genes. Population 1 and population 2 both have 1,000 mice, and each mouse will have 20,000 genes in its genome. However, we could have a total of 50,000 alleles for population 1 and a total of 40,000 alleles for population 2. While the number of genes are the same, the number of alleles could be different. There is more genetic diversity in population 1, for example, more alleles for fur color. There could be more alleles, or there are more alleles for all the genes that are present in population 1. Population 1 is better able to adapt to changing conditions through natural selection. Every population has some genetic diversity, unless that population is made up of clones. Genetic diversity enables natural selection to occur. So let's take a look at what natural selection is. Natural selection and evolution. Random mutations can produce new alleles of a gene. These mutations could be harmful, have no effect, or be beneficial. Let's take a look at an example when they are beneficial. Here's a population of those mice again. Imagine there is a mutation in the gene for fur color to produce a new allele, and that allele, let's say, is a slightly paler shade of brown. If the environment where these mice live has a kind of light brown color, then we have an advantageous fur color in that mouse with the new allele. A beneficial mutation can lead to an increased chance of survival and an increased chance of reproduction. And so the new allele is passed on to offspring. The frequency, in other words, how often it occurs, the frequency of the allele increases within the population. So now we have more mice with that allele. So in general, this process, called natural selection, results in species that are better adapted to their environment. Adaptations can be of various types. To aid survival, adaptations can be anatomical. This relates to physical features. For example, a thick fur and fat layer in polar bears helps them to retain heat in a cold environment. We have physiological adaptations, which are related to internal body processes. For example, desert animals like camels can produce very concentrated urine in small volumes to conserve water. We also have behavioural adaptations, and these are related to actions or behaviours. For example, birds migrate to warmer climates in winter for food and better breeding conditions. So this is genetic diversity and adaptation. Next, let's take a look at something called directional selection, and the example that specification mentions is antibiotic resistance. Populations of bacteria can develop increased resistance to antibiotics over time. We can show and explain this with the use of a graph. So in a population of bacteria, we have antibiotic resistance that might vary from low to mid to high. And we have a number of individuals which have either low or mid or high resistance. The graph for a population of bacteria would look something like this. The bacteria mostly have a mid-level of resistance and a low number of highly resistant individuals. With exposure to antibiotics, the mean number of individuals with mid to high resistance increases because they are more likely to survive in the presence of antibiotics. The graph shifts to look like this. The mean number of individuals with mid to high resistance increases. The graph has shifted to the right. We call this directional selection. Next, we take a look at stabilising selection. The spec wants us to look at birth weight in humans. Initially, there is a wide range of birth weights with lower survival rates at both low and high birth weights. Intermediate birth weights show higher survival rates. So let's have a look at a graph of the birth weights first. We have low to mid to high, and most babies are in the middle range. So we have a high number of individuals that are in the middle, and we have a low number of individuals at the low end and a low number of individuals at the high end. Babies with very low or very high birth weights are less likely to survive. We can add to our graph by looking at something called mortality. Mortality is to do with death rates. We can see at low and high birth weights, there's a high mortality, in other words, a high death rate. Over time, fewer babies are born with very high or very low birth weights. Here's the new graph. We can see there's a much lower number of babies born at low and high birth weights. This is called stabilising selection. There is what we call a selection pressure from both sides. In our graph of directional selection, there was a selection pressure towards the right. Next, we take a look at species and taxonomy. Organisms are classified as the same species if they can mate and produce fertile offspring. Here are two individuals of the same species, and here are their offspring. These are fertile offspring. These can grow and produce their own offspring. However, sometimes individuals from different species can breed, but their offspring are infertile. An example is a horse and a donkey, which can produce an infertile individual called a mule. Courtship behaviours. Courtship behaviour happens before successful mating. Courtship behaviour helps individuals to recognise members of their own species, identify a mate capable of breeding, one that is fertile, synchronise mating so that it occurs when conditions are right for reproduction. Examples of courtship behaviour include bird songs, dance displays, and colour changes. Courtship behaviour improves chances of successful reproduction. The phylogenetic classification system. Classification is useful for identifying and naming organisms. It also helps us to understand evolutionary relationships. A phylogenetic classification system tries to organize species into groups based on their evolutionary origins and relationships. It uses a hierarchy. Smaller groups are placed inside larger ones, and there is no overlap between the groups. Each group is called a taxon. The plural for taxon is taxa, so one taxon, many taxa. Here is an example of a hierarchy that we need to know. It starts off with domain, kingdom, phylum, class, order, family, genus, and species. Unfortunately, we do need to remember the names of these in the correct order. I like to use do keep pond clean or froggy get sick. You can use your own sentence, or you can just remember them in order. Let's take a look at one example. This is Eukarya, Animalia, Chordata, Mammalia, and now it starts getting a bit tricky, Artiodactyla, Balaenopteridae, Balaenoptera, Musculus. Luckily, we don't need to memorize those because I think the pronunciation of those would be tricky for anyone. However, this is a blue whale. Let's take a look at one more. Eukarya, Animalia, Chordata, Mammalia. So far we can see they are in the same groups. However, at this point, we change. We go to primates, Hominidae, Homo, and Sapien. So we can assess and see how closely related these two are. So they're in the same group all the way down to class, but that's when they go off in different groups. This, of course, is humans. We can look at this in a different way, which is sometimes shown in exams. So we can represent Animalia as this large group, and we have smaller groups within groups and none overlap. For our whale, we can see that the groups change after Mammalia. And for our human, we have different groups, smaller groups within groups, nothing overlapping. So this is how we can represent those two species in a different diagram. Let's quickly take a look at one more group. starts off with eukarya and fungi, and you can have a bit of fun trying to pronounce those, but we end up with Saccharomyces cerevisiae. This is yeast. So the final thing to remember here is that the universal way of naming a species uses what's called a binomial system made up of the genus and the species. So if we were talking about our whale, it would be Balaenoptera musculus, that's the genus and the species. For human, it's Homo sapien. And maybe you can have a go at the yeast yourself. The yeast is Saccharomyces cerevisiae. This is the genus and the species. And this is the universal way of naming species using the binomial system. We, of course, have common names for all these different living things: blue whale, human being, and yeast. Next, we're going to take a look at biodiversity. Biodiversity within a community. Biodiversity refers to the range and variety of living organisms in an area. Biodiversity can refer to anything from a small habitat, so we have a log, a pond, a rainforest, a desert, an ocean habitat, or a frozen habitat. We can, of course, go all the way up to the size of planet Earth. Now, the number of different species in a community is called the species richness. A community is all of the populations that might live in an area. We can measure something called the index of diversity. Index of diversity is determined by the relationship between both the number of species in a community and the number of individuals in each species. We have to take both of those things into account. We can calculate an index of diversity, and this is the formula. Now, every question I've seen on this has given this formula, but it is mentioned in the spec, so it might be worth just knowing and remembering it. So let's take a look at what it means. This symbol means the sum of, or the total of, and capital N is the total number of organisms of all species, and lowercase n is the total number of organisms of each species. The bigger the index of diversity, the more biodiversity there is in an area. So let's take a look at an example. The following species and population sizes are found in a desert habitat. The species are listed there. We've got two types of cactus, desert lizard and roadrunner, and we've got the number of individuals of each. We could start by calculating n. That's the total number of organisms. So that would be 50 plus 20 plus 10 plus 5, which is 85. Then we would do n, brackets n minus 1, and n minus 1 would be 84. So that is 85 times 84 equals 7140. Then we would calculate the sum of n times n minus 1 for each species. So for the cactus, there are 50 of them, and n times n minus 1 would be 50 times 50 minus 1, which is the same as 50 times 49, which is 2,450. We do that for the rest of the species. So for the prickly pear cactus, it's 20 times 20 minus 1, which is 380. The desert lizard is 10 times 10 minus 1, which is 90. And for the roadrunner, it's 20. We add all of those together, and that gives us a total of 2,940. We can then substitute into the equation, or the formula, and that would be 7140 from part 1 divided by 2940 from part 2, and that gives us an index of diversity of 2.43. There are no units. You could try one for yourself. A tropical rainforest habitat has the following species and respective population sizes. So here's the information that we need. You have the formula above. So give that a go, and I'll put the answer on the screen in a moment. So here we have the calculations that we need. So hopefully you got those. And we have a final index of diversity. The rainforest shows more biodiversity than the desert because it has a bigger index of diversity. Next, investigating diversity. Genetic diversity is the number of different alleles of genes in a population. We've looked at that before. It relates to variation in DNA sequences between organisms. So diversity and variation can result from variation in DNA sequences. And genetic diversity within or between species can be made by comparing, and these are important to remember, 1, the frequency of measurable or observable characteristics. So for example, if I've got 2 individuals of the same species, we could look at height, mass, body markings, and body proportions. So these are observable characteristics. Or we could look at the base sequence of DNA. So in species 1, for example, we've got this base sequence of DNA for part of a DNA molecule. And in species 2 and species 3, we have these base sequences. How closely related are they, or how diverse are they? Well, we can see that we have just 2 differences between species 1 and 2. That's CT and TG are different. And there's more differences in species 3 in the base sequence. So we can see that species 1 and 2 are closely related and more closely related than they are to species 3. We can use the base sequence of mRNA, which is related to the base sequence of DNA. So species 1, species 2, and species 3. And similarly, we could see how close those base sequences are. So again, we could see similarity between species 1 and 2 is quite close, and different for species 3. So species 1 and 2 are more closely related to each other than they are related to species 3. We can use the amino acid sequence of proteins, and these are coded for by DNA and mRNA, so it makes sense that the amino acid sequence would be related to the species as well. So we've got species 1 with that order of amino acids. This is species 2. This is species 3. And again, we can see there's a difference between those amino acids between 1 and 2 and those amino acids in species 3. So there's only a difference of 2 amino acids between 1 and 2, but there are 3 amino acids that are different between 2 and 3 and 1 and 3. Phylogenetic trees. A phylogenetic tree shows the evolutionary relationship between species. The table below shows the percentage similarity of DNA using DNA analysis between 4 species of bacteria. So here are the 4 species shown in the table, A, B, C, and D, along the top row and down the side. And the first thing to notice is that if we compare A with A, well, that's obviously the same species, so the similarity in DNA is going to be 100%. The same goes with comparing B with B, C with C, and D with D. However, if we compare A with B, we can see that there's an 86% similarity in the DNA. If we compare A to C, there's only a 42% similarity, and A with D, there is a 44% similarity. So this is how you would read the table. And from this, we can make our phylogenetic tree. Usually this represents a very long time, but with bacteria it might not be such a long period of time. However, we can see that A and B are quite closely related by how recently they branched off into 2 separate species. They have an 86% similarity. But we can also see that D and C are very closely related because they have a 42% and 44% difference compared to A, but they are close to each other. And the biggest distance is between A and C. That's the biggest difference in the species. So at point X, point Y, and point Z, we can see that at Y, this is the common ancestor of A and B, and Z is the common ancestor of C and D. X is the common ancestor of all of them, of A, B, C, and D. So that's how you would read a phylogenetic tree. Higher DNA similarity, for example between C and D, indicates a more recent divergence into 2 separate species. So that's it, a whiz through of genetic information, variation and relationships, Unit 4 for AQA A-Level Biology. Thanks for watching. Well done if you got all the way through this epic video. Thanks for watching, and I'll see you soon.